It's a great honor for me to have this opportunity to present our research at WBC Sapporo. Today I'm going to speak about one of our field studies of this title. Mycotoxins are secondary metabolites produced by fungi mold. Mycotoxins cause mycotoxicosis when ingested by the human and animals. Consumption of mycotoxin-contaminated diet may induce acute effects such as the reduced feed intake or reduced production. And recently, exposure of low-level mycotoxin for long period, so-called chronic exposure, seemed to be a major problem under the field conditions. Additionally, the presence of the residues in food products have been also great concern. In the past few years, we have conducted in vivo research focused on the mycotoxin Zearalenone and its metabolites to examine the natural contamination status in farm animal environment, especially cattle herd. Zen is known as a non-steroidal estrogenic mycotoxin, mainly produced by Fusarium species. This picture shows then contaminated rice straws contaminated by Fusarium graminearum. Because of lactone ring of Zen resemble many structural features of the aromatic ring of estradiol, it fit into the binding pocket of the mammalian estrogen receptors and activate it, but can also block it after long-term exposure. Therefore, Zearalenone is assumed to be an endocrine disruptor. So, Zen and its metabolites may exhibit distinct estrogenic and anabolic properties, but also adversely affect the reproductive system of several animal species. After oral intake of Zen, absorption mainly occurs in the intestine, and it gets metabolized mainly in liver, as shown in the figures. After glucuronide conjugations, Zen and its metabolites are secreted with urine and bile. The pathway of Zen, beta-zearalenone or beta-zol, beta-zearalenone, zearalenone, is well understood in case of cattle. In this experiment, we used the previously established method for measuring urinary Zen concentration with LC-MS. At the WBC in Lisbon, we reported for the first time that monitoring urinary concentration of Zen and its metabolites is a useful tool to predict animals' exposure to Zen-contaminated feed on cattle farms. Additionally, the monitoring is a useful method for evaluating the effects of mycotoxin adsorbent, which can be added as a supplement to the dietary feed of cattle. In the present study, we used Difructose anhydride 3, or DFA III, as a test compound. DFA III is non-digestible disaccharide present in commercially roasted chicory. It has already reported that DFA III promotes mineral absorption in rats, humans, and cattle. Additionally, we previously reported that DFA III is useful feed supplement as a prebiotic for the improvement of the health conditions and intestinal microbiota in calves. DFA III is now commercially available in Japan and widely used for preventing milk fever of dairy cattle. Recently, it was reported that direct interaction of oligosaccharide with intestinal epithelial cells and oligosaccharides improve and protect the intestinal barrier integrity and modulate the immunoresponses of epithelial cells using experimental animals as well as pig and poultry. Based on these findings, we developed the hypothesis that administration of DFA III would improve and protect the intestinal barrier integrity in cattle also, thereby allowing to use DFA III as an alternative to conventional mycotoxin adsorbent, particularly in case of chronic low-dose mycotoxin contamination of cattle diet. The objective of this field study is to evaluate the beneficial effects of supplementary DFA III by using our established urinary Zen monitoring system. As a marker of intestinal barrier function, we analyze of serum concentration of calcium in organic phosphate and magnesium. This approach would be evaluate the beneficial effects of DFA III in cattle exposed to mycotoxins. Two Japanese black female cattle herds were used in the present study. They were all of the same age, 9 months old, and similar body weight. In the DFA group, DFA III was supplemented as top dose on the concentrate, totally 40 grams per day. In the other group, no DFA was supplemented and served as a control group. The dose of DFA III was based on our previous report. This details the protocol used for DFA III supplementation and sampling procedures. Urine and blood samples were collected at the start of DFA III in the middle of day 9. On the final day of supplementation of DFA III on day 14, and on the final day of sampling, day 23. As shown in the picture, the urine samples were collected using the massage method. This slide shows the procedure used to measure Zen concentration by LC-MS. Briefly, 0.5 ml of each of the urine samples was pre-incubated overnight with buffer and enzyme solution. After extraction with column, the eluted sample were injected injected and analyzed by LC-MS. In parallel, the urine creatinine concentration was measured to correct for the different urine values, and finally, Zen concentrations was determined and expressed as the creatinine correction concentrations using this formula. A measurement of serum mineral concentrations were conducted using auto-analyzer. This slide shows the result for the analysis of urine concentrations of Zen, its metabolite is alpha-zole, beta-zole, and total of the Zen concentrations during experiment period with DFA III and control. As you can see, data shows that the treated animals had significantly less Zen concentration in urine, indicating a reduced absorption from intestine. This slide shows the result for the concentration of serum PI calcium magnesium during the experiment period with DFA III and control group. No significant difference could be observed for most of the time points, but a trend to slightly higher levels could be noted, with a significant difference for PI at day 23. In the intestinal epithelial cells, there are two transport pathways, including the transcellular transport that is facilitating the absorption of nutrients, such as amino acid and sugars, and the paracellular transport, which needs to be as low as possible, as by this route, toxins and even pathogenic bacteria can enter the body via this route. Based on the present result, we assume that the direct effect of DFA III on intestinal barrier integrity, particularly on the tight junctions, may be similar to galacto-oligosaccharides as previously reported, thereby reducing mycotoxin absorption and preventing an intestinal inflammation reaction as described for many mycotoxins. Together with previous shown beneficial effects of DFA III on the intestinal microbiota, we conclude that the DFA III may be an entering substance to be added to feed to improve animal health and well-being in the intestinal tissue. Finally, we conclude as follows. Firstly, DFA III supplementation to diet feed alters zinc and iron, PI, absorption level in cattle. Our result, the first indication that DFA III supplementation may reduce the level of mycotoxins that reach systemic circulation and are secreted in the urine. These results also confirm the value of monitoring urinary mycotoxin concentrations to demonstrate the prevention of mycotoxin absorption. Moreover, DFA III seems to be able to improve tight junction dependent intestinal barrier function. In consideration of these dual activities, we recommend further field studies as well as creation of a database for the assessment of DFA III as a direct supplementation, as a dietary supplementation to reduce mycotoxin absorption and mitigate adverse effects of mycotoxin in cattle and other animal species. Thank you for your attention.